Sounds working.
Well, I had a little music to start.
Wait. Just I want to do a general presentation, please.
Yeah.
So welcome everyone. Are we online?
We're online.
Yes, we're online. So welcome everyone to all the students assembled here today. And to anybody joining us online for this this first session of the 2024 IMBRC Thesis Defense. Oh, sorry, I'm having myself in the feedback loop because I forgot to turn my sound. So we have 3 presentations per session. We will have students who will have 15 minutes to present, as you know, and then we'll have about a 15-minute discussion. When you get to about, when you have 3 minutes left, I will wave my hand, and so just try to keep a lookout for that. If you keep talking and you have reached the 15-minute mark, I will stand up. And that means you need to wrap up instantly and stop. And so yeah, we'll have the members of the jury will present each other when we start the discussion. And so thank you. We'll start with Francine Soula, will present us with our thesis work.
Okay. Hi everyone, thank you for coming. And today I'm here to tell you a story, a friendship story between the black mangrove Avicennia germinans and the sea purslane Sisilium portulacastrum. My name is Francine Soula, and this thesis was supervised by Mark Hodgson from Edinburgh Napier University in partnership with the Sankandi Youth Development Association in the Gambia. So, to set the context of this research, I would like to invite you to travel with me to the Gambia for a few seconds. And so, if you feel comfortable, you can close your eyes and let me guide you through this journey. So, it is the morning in West Africa. You just woke up in the village of Sankandi, and the birds are singing. The sun is rising. It is already very warm. It's 30 degrees in the morning, but today you're really excited because you're gonna head to the mangroves. So after a 20 minutes drive in the bush, you will finally arrive in the mangroves. And you can open your eyes, and this is what you see. It might not be the mangroves as you imagined. It's actually very large mudflats, saline and arid. There is no vegetation, unfortunately, because everything died in the 1970s due to a drought and unsustainable harvesting by the local population. So as you can see, there are dead tree trunks. These are Avicennia germinans. So the local population now doesn't have the vital resources that the mangroves were providing, so wood for timber and fish in the river. But now the local association, the Sankandi Youth Development Association, is trying to restore these mudflats, and they just planted trees, as you can see here in the video. And our goal as researchers is to help them to expand the restoration activities to the wider mudflats. So we came across this interaction between species on the mudflats, which was really interesting, and we wanted to investigate further. The sea purslane seemed to help the Avicennia germinans, the black mangrove, to grow, and this is called interspecific facilitation, in which one species helps the other to survive and to establish. We also wanted to investigate, I don't know what's happening. It's showing up here, but not on the screen.
That's strange.
Any problem?
Yeah.
I know, the screenshot. Maybe it's better if you let the door open for the air and for problems. What's the problem now?
The screen just shut down. 2 minutes and a half in.
It was not like that today. You have to put the HDMI soon. Yeah. Just wait. So the special thing for you.
Yeah. Okay, well,
Just
let's
go back
keep
to where we
going.
were.
Yeah. So it was this slide. So so I was telling you about the interspecific facilitation, in which one species, the sea purslines, helps the other to survive. We also wanted to investigate intraspecific facilitation, in which individuals of the same species help each other to survive, focusing on the black mangrove. So this is our target species. So the aim of this study was to investigate the facilitation processes that I mentioned between these two species to help the restoration activities in the mudflats of Saint-Candide. This story has 3 chapters. The first one is a survey of the association between the sea purslane and the Avicennia germinans. The second chapter is a transplantation experiment of Avicennia germinans seedlings. And the third chapter is the one I'm the most excited about, the transplantation experiment of sea purslane. So this was never done before, and that's why I was really excited to try it in the mudflats, and you will see that we got unexpected results. So first, starting with this association, we wanted to investigate how sea purslane would act as a nurse species for the Avicennia germinans seedlings. So we chose randomly and selected 12 plots across the site, and each plot was 15 meters by 15 meters. Half of it was covered by sea purslane, and the other half was bare ground. So we can see here, in is inside sea purslane, out is the bare ground. And we measured a range of parameters within and outside. The tree density, Avicennia tree density, the surface moisture, surface salinity, pH, clay content, organic carbon, and finally, temperature. And this was the statistical test we used was t-tests, and these are the results that we got. Very simple analysis. The first one I want to focus on is the tree density. So you can see it was much higher as we expected in the sea purslane. On the bare ground, it was almost 0. Then surface salinity was much lower within the sea purslane, meaning that this plant was lowering the salinity of the soil, and that was a significant interaction. And finally, the surface moisture was much higher in the sea purslane patches, meaning that the canopy was actually retaining the moisture compared to the bare ground when it was drying very fast. We got a negligible variation in pH, no significant interaction in clay content and organic carbon, so I'm not going to focus on them. But something that is not shown here is the temperature. I measured temperature in the sea purslane, and it was much lower compared to the bare ground. It was reaching up to 56 degrees very frequently in the bare ground, when it was lower in the sea purslane. It was reaching up to 50 degrees. So the significant difference in temperature was huge. And so this is the end of the first chapter. Now let's move on to the second, the transplantation experiment of Avicennia seedlings. We had 3 blocks across the site, and in each block we had 3 treatments. So the first one was planted on the bare ground, 4 seedlings of Avicennia per meter squared, and that was the sparse treatment. Then we had the dense treatment to measure how planting density would affect the survival of the seedlings. The dense treatment was 11 seedlings per meter squared, and we had the last treatment planting 11 seedlings again per meter squared, but within a sea purslane patch that was already occurring on the mudflats. So we could see the effect of this plant on the survival. Over 2 months, we measured the survival of the species, of the seedlings, and we carried a survival analysis. And these are the results that we got. We got actually 94% mortality, very low survival. But there was interesting trends still in this survival. The lower curve is the sparse treatment. So it's the lowest, meaning that the survival was the lowest and the seedlings were dying faster. And in the dense treatment, there was 20% more chances to survive compared to the sparse treatment. And then that's the orange curve. And the significant difference that we found was within the sea purslane, there was actually 24% more chance that the seedlings would survive compared to the dense treatment, meaning that the sea purslane actually had a huge effect on survival. And finally, the last experiment. the sea purslane transplantation experiment. We found this site on the mudflats, separated by a creek, as you can see here. One side was full of vegetation, Avicennia was regenerating, and sea purslane was thriving. The other side was void of any vegetation. So we thought, what if we could transplant sea purslane from the growing side to the non-growing side and see how it would survive if it was going to establish or not. So it shows 3 distances. The close distance is close to the creek, so high moisture and low salinity. And then as we went further from the creek, it was increasing the water deficit and the salinity. And we had 4 patches per treatment, so 4 replicates of about 50 centimeters by 50 centimeters, this size of patch. Then over 2 months again, we measured how these patches were doing. We had 2 methods. The first one was to measure the number of leaves per shoot. So we counted the number of leaves in some selected shoots, randomly selected, that we tagged, and then every week we're gonna come back and count the number of leaves. But this method kind of had an issue, as you will see in the future. So we had another method. It was the health scale. So I created a health scale going from 0 to 4 and describing the health of the transplant. 0 is a dead patch and 4 is a patch in excellent health. And so every week we're going back and taking pictures of these transplants, and then we could assess the health with the number. These are the pictures how it looked in the beginning, so very healthy patches. You can see the labels that we used for counting the number of leaves. And at the end, this is the result. Cows came and they ate our plants. So this is what it looked like at the end. No leaves anymore because of the cows. So we couldn't use this method, and we actually used the health scale until the end of the experiment. This happened in the far and middle distances, but in the close distance, it was too close to the water for the cows to come. So actually, some patches were actually doing great. The health was still declining, but they seemed to have established, and they didn't lose all their leaves. This is what you can see here, the 2 methods. So declining health over time. The number of leaves went to 0 for the far and middle treatment, but the close was still higher up because not grazed by the cows. And then same pattern in the health scale, so it was a bit higher in the close treatment. That treatment was actually declining in health as well because of the tides. So some patches were just submerged in high tides, and that was just ripping off some leaves and shoots. So that was also an issue that contributed to declining the health. So this is the end of the story. Let me wrap up and remind you what I said in the past. So we actually showed the nursing effect of sea purslane. It was decreasing the temperature of the soil, decreasing the salinity, and increasing the water moisture because of the canopy effect. So the canopy was actually decreasing the evaporation, and that contributed to decrease the soil salinity. Another effect that the plant could have was it is it is a succulent plant, and it has a halophytic capacity, so meaning that it can absorb salts in its leaves, and that also contributes to lowering the salinity of the soil. So all of this explains why there are higher Avicennia tree density in the sea purslane compared to the bare ground. Another effect that was not studied here is the trapping effect. So the canopy of the sea purslane could trap the Avicennia seeds that would just be washed away in general, but because of the canopy, it traps the Avicennia seeds, and that could explain why there are higher densities within the sea purslane. This was not proven here, but further experiment could just investigate this effect. Another suggestion is to monitor other soil parameters that the sea purslane could impact. So for example, the redox potential, oxygenation of the soil, and its porosity, for example, and that could also explain why Avicennia are thriving more within these plants. I wanted to explain why we got such a high mortality in our Avicennia seedlings. Oh,
I
yeah.
can't say.
Sorry.
Okay.
So-
thank you.
Actually, these seedlings were exposed to the sunlight all day, and as I said, it was 56 degrees on the bare ground, so very high, and the seedlings did not survive these harsh conditions. And also, with the hypersalinity of the soil, it was too harsh for them to survive. So, a suggestion would be to plant in the rainy season. There you have more fresh water, and the seedlings don't have the stress of the hypersalinity, at least it's lowered. It's lower temperatures on the surface, so all of this could help the survival, and this is another further experiment that we could do. Another consideration was the transplantation stress. So these seedlings came from another site, another mangrove healthy site, but they were grown in sandy soil, and the mudflats were actually very rich in clay. So this difference in soil texture was a bit of a problem, and that might have contributed to the very high mortality. So in the future, we could adapt the seedlings to the soil before, create a nursery, for example, in the mudflats, with seeds growing directly in the soil and not being that stressed by the change in environment. We would also need to investigate further the intraspecific facilitation. So, as I showed you, the sparse and dense treatment didn't have a significant difference. Still, we found a trend, so this has to be investigated further. And then finally, we need to reassess the health of sea purslane and its response to transplantation, because as you saw, the cows came, so we would need to protect them from herbivory by creating fences around the transplants. We could also plant them in the rainy season, because as for the Avicennia seedlings, it could lower the stress from water deficit and hypersalinity. We could also use bigger patches, because the patches we used were pretty small, and when we were moving them around, we cut the roots and the shoots, and that was disturbing the transplants. So if we use bigger ones, maybe the center of the patch could remain healthy and then established better in the future. This is it with my story. Thank you. And I made a little video of my project, so if you want to have a look at it, you can scan this code and it will show you the video.
Thank you very much, Francine. If you want to, it's
Yeah.
better for all of you if you stand on the other side of the screen and use it to move, so people online see you. Because otherwise, if you're moving out of the screen here, people online cannot see you. And I think you have a few friends and maybe family members watching online.
Yeah.
So I'm gonna give the floor to Fabio first.
Okay.
Fabio from Ancona.
Look at the very nice piece of work, Francine. Well, I hope that the course of restoration that you could make in some way help. Of course, here we're talking about different organisms. But no, to me, this is very interesting. Look, I think once again it confirms something that I told to the people who take the course of restoration with me, that obviously maintaining and conserving is always easier and less expensive and less laborious than trying to restore. Obviously, this is a situation in which it is very clear that really the mangroves and the porcelain, obviously their presence itself modifies the texture of the ground and makes it suitable, which is basically the same thing that the seagrasses do in the sea. Now, about this fun thing, I wonder if besides, I agree with your suggestions here, all these things to me they make sense. A couple of points. Now, I have to admit I'm not familiar with the biology of the Avicennia. Is it is this species reproductive all the time during the year? So for example, is it actually feasible to do the transplants of the seedlings even during the rainy season?
Yeah, so actually the species is flowering in March, so we're there until March. And then the seed starts to spread in April. And then I think they settle in April, June, just before the rainy season that starts in June. And then the seedlings can grow actually very well during the rainy season because it lowers the salinity and everything.
Okay.
So we didn't have this chance to be there at the right moment at the right timing for this. But I think, yeah, in the future, if we're to do it, planting in the rainy season and following the species flowering would really help the survival.
And then look at the other suggestion I have, but this I'm not sure if it is actually feasible or not. It might be okay in the plots where the transplants try to modify the texture of the ground in some way by adding some. I'm not sure, in Italian we call it humus, which is the superficial the soil. Trying to collect a bit from the mangrove forest or the other places where there is this soil with a higher organic content and try to mix it with the clay, the soil in the plots where one wants to do the transplants.
Yeah, that could be interesting to do because we created a nursery actually there. So the goal is to plant in a nursery and then be able to transplant these seedlings. So within the nursery we could modify the soil conditions. And what we did for now was to put some hay on the soil, so like just dead plants so that it would create a shadow effect, kind of like the sea purslane, but just by cutting herbs on the side and placing them in the nursery. So that also helps for the seeds to survive better. Yeah.
And then another couple of quick questions. One is when the cows went there and grazed the plant, I see the leaves disappeared, but do you know if the stems underneath were still alive? Would they be able to put out the new leaves?
Yeah, so we actually kept monitoring these plants after they got grazed, and the stems were still looking kind of healthy. They were still like yellow, which is the color they were in before. And some transplants had some small leaves starting to regrow, but it was very slow. And we actually then started to count the total number of leaves on the transplant, but this was kind of remaining stable because I think it was not the right season for them to start thriving again because it was stressed by the salt and the lack of water. But maybe if it had water in it, the plant would recover and start growing. But that we couldn't see, and we were there for a short amount of time. So yeah, but I had hope. I loved them, and I thought they were really cool, and I think they could survive better.
Okay.
So I think we really need to do further experiment on that.
Okay, and then the very last point, are now the local populations aware of the importance of mangroves, and are they beginning to realize that they shouldn't send cows they graze there?
Yeah, the cows are just free range, they just go around everywhere, you don't really control them. So, yeah, I didn't really emphasize the fact that the cows came to eat, and I think they also need to eat the cows because they go to the mangroves and that's the only place where they can have fresh herbs because in the dry season it's just so dry everywhere. So yeah, it's a bit tricky, but they do realize the importance of mangroves in general, and if they realize that this plant is actually the key for their mangrove to recover, they might do something. Yeah.
Okay. Very good. Good job. Congratulations.
Thank you.
Thank you, Fabio. Gauthier?
Yeah, congratulations first on a very nice presentation. I really like the very creative way to introduce your topic, and it's a very good discussion in the end. So I have a first question about this person transplantation experiment. Sorry. So what was the big idea behind that?
Okay, so the big idea was maybe if that plant was actually establishing and starting to spread quickly or easily enough, that could be a way to help the restoration of Abyssinia germinans. So just putting transplants across in the mudflats, just in random places, could create like small patches of natural regeneration of Abyssinia. So with having seeds coming and just regenerating in those patches.
Okay, and in the light of those objectives and regardless of those cow events and all problems you had,
Yeah.
what are your conclusions about it?
Well, I think it was not growing as good as we would expect. Like I was thinking in the best situation, it could just spread really quick, and then we would have really great results, and that could be used in the future. But now with this conclusion, in that it doesn't work in the dry season, and we would really need to adapt in the rainy season, and with the cows also, because it is an important consideration as well. It might be difficult to use this method, at least for now. Or maybe we could create a nursery as well, where it is protected, yeah.
Okay.
And create this nursery, and then transplant some patches and.
Okay, and can you please go back to this slide where you showed this experimental design for the third chapter?
Yes.
The aerial view.
Mhm.
Yeah, this one. So it looks like it's very quite densely vegetated on the left, and there's nothing on the right. Do you have an idea why this is like that?
I might have an idea in that the cows don't have access to this place. Oof, sorry.
You can just swipe.
Oh yeah. But also maybe the sea porcelain was there, and then it actually created very thriving meadows that were a bit elevated above the ground, and this elevation was actually making them not flooded in the very high tides, and that was helping them not to be washed away and everything. While on this side, first there was the cows that could come and eat the plants here, and here they would be just washed away by the tides. So I think this ecosystem is just recreating, and it's just increasing, because at the start there was the porcelain, and then the Abyssinia came and it started increasing again. But since there's nothing at all, I think that's why it's not growing. So there might be an elevation.
It's difficult to initiate restoration.
Yeah, it is tricky.
In these conditions where everything has appeared, like that's one of the things I say during my course. I mean, it's always better to be preserved and conserved than to be forced to restore.
Okay, one last question if I still have time. You mentioned several possible explanations underlying the mechanisms underlying this facilitation between the porcelain and the mangrove tree, the Abyssinia.
Mhm.
If you were given 6 more months and infinite money, and with the objective to identify the main ecological mechanism underlying this facilitation, what would be your strategy?
I would consider other parameters probably. So as I mentioned, the redox conditions and porosity, and continue this analysis of the soil conditions.
Yeah, but how would you be able to disentangle between like the seed trapping, the shading, or all the possible mechanisms that make sea porcelain facilitate Abyssinia?
So
You can measure as many variables as you want.
yeah.
It's not gonna help you.
So I would do other experiments, because I think by experimenting that's how we know. For
Yeah.
example, for the seed trapping, I would design an experiment in which I have a sea porcelain. I actually thought about it when I was there, but I didn't have time to do it. So I would have a sea porcelain patch here, and then the bare ground, and maybe line up some seeds and see after like a few days, count how many seeds are there in the patch of sea porcelain and how many are just washed away. So that would investigate the seed trapping experiment. And then for the shading effects, we could also have a treatment without sea porcelain, but as I mentioned for the nursery, just like herbs that we cut and we put like hay, we put on the ground to create this shading effect, but without having the modification of the soil necessarily. So having different treatments like that and seeing the survival again of the seedlings.
Okay.
Yeah.
Thank you very much.
Alright.
Congratulations again.
Thank you.
Thank you very much. Alright, we're gonna set up
Thank you.
for our next presentation.
Yeah. Ah, no. I do, oh, no, it's fine, it's fine.
Okay.
No, yeah.
Okay.
Alright, so Anna, you're up next to present us with your work. Probably it's 15 minutes.
So, good morning, everyone. My name is Anna Almospin, and I'm going to present you my master's thesis with the name First Description of the Sediment Bacteria Community adjacent to healthy coral heads within the lagoon of One Tree Island Reef. And my supervisors were Dr. Graham Clark, Dr. Steph Gardner, and Jakob Quadri from the University of Sydney. So first, One Tree Island Reef is is a coral reef ecosystem in the southern region of the Great Barrier Reef. And considering all the past coral mass bleaching events, 5 and 7 years, predictions suggest that the southern region will be more vulnerable to climate change, and One Tree Island will be at even higher risk because it's a lagunar reef. So why is this? The seawater inside of the lagoon stays enclosed during low tides, and it has no inflow from the cooler out lagoon waters. And during extreme heat events, the seawater inside of the lagoon can reach extreme temperatures, and this can trigger coral bleaching. And so, there's a high interest in studying the coral head inside of the lagoon, but to understand and protect the coral reef, we have to consider all the components of a coral reef and how they contribute to the stability and function of the entire ecosystem. Reef health assessment normally focus on the coral and the algae cover, and also on the diversity of the associated fauna, but they tend to overlook the sediment. And actually, sediments are produced in situ by erosion, bioerosion of the coral, and this makes the sediment permeable and also bioactive. And then the sediment is also the reef compartment with the most diverse microbiome, not only compositional but also functional, and in particular the bacteria community. And sediment bacteria are significantly contributing to the composition of organic matter and also to the recycling of nutrients. So they are actually actively supporting the coral reef ecosystem. In addition, the bacteria community is a good indicator of environmental perturbations, and also there's studies that say there is a direct interaction between corals and the bacteria community, but the lack of knowledge about the sediment bacteria community makes it really hard to study this link. So with this in mind, the aim of this study was to gain knowledge on the composition and distribution of the bacteria community of sediments adjacent to healthy coral heads inside of the lagoon of One Tree Island Reef. And the main questions were what was the composition and if it varied across the lagoon, and also if there's all the factors, environmental or coral related, that can influence this composition. So how did we approach this? First, during the field trip in Austria Spring 2023, 21 coral heads from the genus Varieties were randomly selected within the lagoon. And these coral heads were incubated in situ to measure the respiration and the gross primary production. And we calculated their ratio as an indicator of the coral's trophic state. We also took photos and did a 3D model of the coral to calculate the surface area. And we also took sediment cores to analyze the grain size and the nutrients content of the sediment. All these factors were used as predictors in the statistical analysis. For the DNA, we sampled sediment inside of the chamber. We homogenized the sample and subsampled it into 3 replicates, from where we extracted the DNA. And then we amplified the hyperviral region V3 to V4 of the 16S ribosomal RNN gene. And we sent this PCR product for sequencing. So once we had the sequences, they were treated in R. And first, we eliminated all the runner sequences with the packaged data tool. And we also inferred the amplicon sequence variants, and amplicon sequence variants are a group of sequences that differ from other sequences at the level of one single nucleotide. So they basically are taxonomic units. And we calculated the abundance of each ASV per sample. And we also assigned their taxonomy using the genome taxonomic database from the University of Queensland. So for the data exploration and the statistical analysis, I use the microecho package that also includes other functions from other packages as speed and ILD6. So to answer which bacteria compose the community, I calculate the relative abundance at phylum level. And here you see 5 of the 21 samples. But almost all the samples had the same phylum composition as these 3, where Protobacteria were the most abundant, followed by Bacteriodota, Actinobacteriodota, and Planktomycetota. And these phylums are known to be present and abundant in sediments near reefs. But sample 4 and sample 3 varied. So to check, verify if this was a natural distinction, we checked the phylum also in every replicate, and as you can see, it's really clear that sample 3 has a replicate that was an outlier, and inside of this Firmicutes, it's a family, the Amphibacillus is highly abundant, and it's a facultative anaerobe, so maybe the replicate was contaminated, but we keep it as we only have 3 replicates, and sample 4, here, it's a natural distinction, like there was no problem. So now to know if the composition varied within the location, we conduct our principal coordinate analysis using the weighted unifrac distance. And here in the plot, you can see that almost all the locations share a similar community, and with the exception of sample 4. And sample 4 is located really near to the channel that communicates the inside of the lagoon with the outside of the lagoon. So maybe this can be the reason why the community is different, but it was not the only sample site near this channel. So we thought that other factors were actually driving these differences. So to see if other factors were driving it, we conduct a distance-based redundancy analysis using also the weighted unifrac distance. And here, by the length of the arrows, you can infer that the gravel and the nitrogen content were the main drivers of the difference in community, and this was supported by the mantle test. that also told us that the sorting level of the sediment was responsible for the distinctions. And again, sample 4 is apart from the main cluster. And so I selected sample 4 and what seems to be the sample that differs the most of it, sample 20, to kind of compare the environment conditions. And in sample 4, the sediment has a low mud content, so maybe the sediment is more permeable, and this would explain the low nitrogen content, as it's easily lost to the water column. And conversely, like site 20 has a lower mud content and also a nitrogen content, and this suggests that the sediment was less permeable, so the nitrogen can be accumulated in the sediment. But site 20 is also the site that has most gravel content. But if you may see in the picture, almost all the sediment, all the gravel, is on top of the sediment, not within, and we saw it also in the course of sediment. So this site was really near to a reef that was highly eroded. Okay, and let us take a closer look. So which taxa are contributing to these differences? And for this, we did a differential abundance analysis using the RDX2 package, and we visualized it with a heat map. And so the results show that out of 1,000 families, mainly 6 were contributing to the difference. And I selected the 2 that stand out the most, so Rhodobacteracea, because it's highly abundant in almost all the samples, and also Alteromonodacea, because it's only highly abundant in the sample 4. So let's check what these bugs can do. And the family Rhodobacteracea is widespread in the marine habitats, and they are known to be involved in biogeochemical cyclings from sulfur and carbon. And they comprise not only symbiotic but also pathogenic bacteria, and this is interesting because our recent study suggested that pathogenic bacteria from this family are associated with the stony coral tissue loss disease. So it's very important if the corals are vulnerable. And also, then in the family Alteromonodacea, we find the genus Pseudoalteromonas that has a relative abundance of 46% in the sample 4. And Pseudoalteromonas are widespread also in the sediments and found in sediments associated to coral reefs. And they are suggested to be key in the composition of particulate organic matter and also in the nitrogen cycle. And in addition, there are studies that suggest that it can influence the settling and the metamorphosis and success of invertebrate larvae, including also coral larvae. So it's really interesting to see them close to corals. So what can we conclude from this study? First, sediments close to healthy corals of the genus variety show similar bacteria composition, regardless of the location within the lagoon of One Tree Island Reef. Second, the bacteria community can be predicted by environmental factors, specifically like the amount of gravel and nitrogen content, and also the sorting level of the sediment. And then, although we didn't saw the factors, the coral-related factors contributing to the difference, a high amount of bacteria related to the decomposition of organic matter may suggest that there's a high bioactivity around corals. And yeah, all in all, this study potentially aids in the monitoring the health of One Tree's coral reef. And related to this, it can serve as a baseline to identify possible shifts in the bacteria community linked to environmental perturbations.
and I got a minute and a half to go, you could have taken more time. But
Okay.
that is fine, it's better than to go over time. So I don't know which of you gentlemen wants to go first?
I can start.
Alright.
Is that the picture from your fieldwork?
Yeah.
Okay. I have totally different idea of Australia.
That's Queensland, that's that warm humid part of the country.
No, it was actually like it was really weird the weather that we had. Like it was not what they are used to, and they were like, you will see like perfect weather, sunny, and I was like, it's raining when I was there, and it's hard.
I'm literally feeling home in Brittany. I have maybe a first question about the sampling design. What was rational behind? I mean, where did you choose to sample here and there in the lagoon?
Like it was actually because they were at the same time there was another research, and they were researching, like they were doing real life surveying of the diversity of the community associated to the coral reefs, like fish and everything. So they actually chose like the same areas to relate their conclusions with what we saw, kind of. Like, yeah, I mean,
So what was the initial assumptions? Normally when you choose a sampling design, it's because you expect to see the influence of some environmental variables, and you mentioned the presence of the channel, so it makes a lot of sense, of course, to sample next to the channel. My question is more about the potential drivers of variability within the lagoon. What would they be, in your opinion?
I mean, the current is not, like the current inside the lagoon is not uniform, like it changes, it's like, can I go back?
Yeah, sure. You had a map.
Oh yeah, sorry. Like the currents are actually like they move like this.
Just to show you that you can take the slides and not stand in between us and the screen.
Really try the next one to stand on the other side.
So they move kind of in circle, and they accumulate here. And like this study that also the group is doing that says that the nutrients are kind of accumulated in this area. I mean, for me, it's what I actually like got. Like I didn't participate in this sample, but it was more like they wanted to have kind of a view of the whole lagoon. And it's because like it was the first time someone was doing this. They actually wanted to have kind of a view of all the lagoons.
Okay, and if I understand well, it's a specific lagoon in the sense that water is basically entrapped in it with very little renewal.
No, like it's like it has like a water pump that it's moving like the water constantly, and it's like we only incubated them for 2 hours.
Yeah.
So.
No, but I mean the lagoon.
The lagoon.
The lagoon has basically trapped water in there.
Yeah.
So the water is subjected to lots of variability, probably with the temperature, salinity, maybe concerning the weather you had.
Yeah.
And you say that the fact that the water is entrapped in the lagoon can cause some bleaching. Did you notice some bleaching?
Did you
go on the field?
Yeah, sorry, this was done like before.
Yeah.
I went to the second time.
Okay, yeah.
Like because they did like a resampling to compare, and my second time because when they did this sampling there was no coral, like the coral bleaching event that occurred that year didn't affect One Tree Island. Yeah. But the second time when we went, it affected like the temperatures, like the high temperatures reached One Tree, and you could see how the corals were bleaching.
Because paradoxically, sometimes those areas that are naturally hypervariable because the water is entrapped are actually where you can expect to see the most resistant population because they're used to it. So you would actually expect to see less bleaching in those areas.
No, yeah,
than
yeah,
in others.
yeah. Like people, like for me, it was like this.
Yeah.
I've never been in a reef, and they told me that once there's like a really coral mass bleaching event,
Mhm.
like when it's confirmed, it's because it's actually also affected corals that you wouldn't think they will affect it, and like this waters when we went, like 1 week before they had reached like the 33 degrees inside. So for them, like this, like really a limit for the corals, for all kind of corals. Like it's really extreme temperatures.
So maybe a last question about your sample form. I'm still not sure. Do you consider this is contamination? Do you consider your results are reliable? Seems like an outlier. Really?
Okay.
No?
Like I mean, I don't know.
The sample 4.
It's us. Sample
Three.
4.
Was it 3?
Three,
What's
three.
sample 3? Sorry.
Yeah.
Right. Which clearly stands out.
Yeah. I mean,
Do
it's
you
not.
think-
No. Sample 4 was the one standing out.
Yeah, that's what I said. Sample
Yeah,
4.
replicated sample 3.
No. Yeah, the replicate
Yeah.
that was an outlier was in sample 3, not in sample 4.
Okay. So that's it.
Right.
Thank you.
Fabio, thank you, Gauthier.
Okay, thanks. Okay, nice work. Okay, well done. Look at a couple of things that perhaps I missed during your presentation. What was more or less the distance between the different corals that you sampled?
Like almost all were like between the 10 meters, like 5 and 10 meters from the.
Okay, yeah. I mean, between approximately
Yeah.
between a coral and okay. See. And then what was the distance of the spot from which you picked the sediment from the
It's
coral?
really close. It was like inside of because we put first the chamber, so we sampled
Okay,
inside, so it
within
was
the-
really
the
close to the
chamber.
coral.
Okay,
Yeah.
okay, see. Okay, look at okay, we're talking about an area for which I cannot call myself an expert. But it seems to me that in the end there was okay, apart for that particular situation, not a lot of the spatial variation in the end. And
No,
the-
I mean it's true that this study was kind of like really a baseline, like because there was no.
Look,
Like they really went when there's no coral
look
bleaching.
at I'm not
Yeah.
okay. This is not a criticism. And they look at, I mean, the studies of the spatial variation like this are always very, very valuable. I mean, they are the premise and then to do other types of the work. And look at what I did was very useful, then very valuable. I'm not criticizing that. Look at actually. Another point that I noticed, now among the groups that you recorded, I didn't see any cyanobacteria. Now I was wondering, is that because they used a marker, a part
Yeah, like
of
the
the bacterial
16,
genome that doesn't amplify
Yeah, this-
cyanobacteria? Or
Yeah, the 16 is S ribosome is actually for bacteria. So the cyanobacteria, they are like.
Okay.
Yeah.
no,
Plankton,
because
so they
in
are
a
not.
tropical area like that, I would
Yeah.
expect also.
Yeah,
some
of course, like this, like
cyanobacteria
you
there.
can really do more study. Like you can see the cyanobacteria. I mean, there are studies that actually show that inside of the sediment you find cyanobacteria, and they say now that maybe it's like when the coral bleach, they stay inside of the sediment and they are mobile, so they can go back. And, I mean, it would be inside of sediment. They decided back.
And then they look at another point, is okay, you mentioned that Rhodobacteriaceae were one of the main group. So you mentioned that if I understand correctly, that they are associated with the pathogenic situations.
Yeah, some
or-
of the.
some, okay, not from this point of view, again, a question from someone who is not an expert in this area. Okay, do you think in these situations, I mean, what's the case, maybe if there is a disease in the coral, will actually be caused by the bacteria, or maybe simply because the coral is unhealthy state, then that particular type of bacteria will tend to
Yeah, I mean,
proliferate.
Yeah, like corals, when they are really stressed, like they produce this whole, like a lot of mucus. So they say that that's kind of the protection for the corals in that moment because they are really vulnerable, like to protect them at that state, like from vulnerability. But like there's a moment if the sediment, like because the bacteria sediment, like when to reach the coral, the sediment has to be rest suspended, or normally like that's the transit, like the way, the pathway. So if they reach the coral at that moment where actually like all the mucus doesn't stay on the coral but it's excreted,
Okay.
so then like the pathogenic bacteria can affect, or can, like it's not that they trigger the disease, but they are associated
Okay.
with this. So it's like
That's, thank you.
All right, well, we have time, so I'm going to take the opportunity to ask you a few questions, and thank you for the presentation. I wonder, what do you think about, how strong is the link between the abundances you observe with powerful techniques such as using DNA, like PCR and sequencing compared to the actual abundances in the community? What do you think about that? Is it a direct, strong link, or is something maybe interfering with that in a way?
I mean, there's always like, we know that all these methods that are used have kind of bias that can introduce errors into the sequences, and that's why like we use this Data2 package that actually like you can delete what you would, as like I had as a controls for the PCR and for the extraction of DNA, where
Okay.
you can have a lot of erroneous sequences, and so I use this the bacteria that appeared in contaminants and in blanks to actually like clean my data set.
Mhm.
And there's like an erroneous like is this the question I'm answering?
Well, I'm wondering because you have amplification and then you have sequencing, and sometimes you can be worried that the abundances you get are not directly representative of the ones in the community, because maybe some sequences are more
it's true.
easily amplified, so you
Yeah.
get more copies,
Okay,
and then
understand.
that was
Yeah,
the
I mean,
sense of the
and
question.
it's actually also that I looked at the total bacterial community.
Mhm.
So, and this kind of analysis doesn't tell you what's actually inside, like at that moment.
Yeah.
So for that you have to see the active bacterial community. So you have to extract RNA, and then from there like do the amplification, like the complement DNAs and so on. So it's not like maybe I find a bacteria that is actually not present there.
Okay. Another question before you go on. One thing that was not totally clear to me was what was your part of the work? You didn't do the sampling for that. You did another one, but then did you do the lab work?
Yeah.
All the lab work
Yeah,
and all the
it's
data
fine.
analysis is
Yeah.
yours. Okay, so it's a lot of work. A technical question: why would you do mental tests after an RDA?
I mean, I tried to to do a comparison. Like what I learned was the simple analysis, that it's between two groups,
Yeah.
and I thought like to do it with all, but it was kind of messy for me. So I used the Manti test because I think like I'm not really an expert in statistics.
That's fine.
I went crazy with this. But like Manti test allows you to compare like more than two groups.
Yeah, but actually in your
Was it so?
RDA you have a multivariate regression model already. But if you want, we can discuss it.
Yeah,
If
okay.
you want to go back and make that a little
Yeah,
cleaner, I think it'd be more interesting. And it could allow you to actually tackle some of the things Gauthier was mentioning about the spatial relationship between your station,
Yeah.
and you could include that explicitly actually in the modeling, either just the distance between your points or the actually taking account to the recurrence and how
Okay.
how the stations might be connected in terms of hydrodynamics, so that could be interesting if you wanna have a chat about that.
Yeah, that would be great.
Any questions from the audience or online? No, but you have congratulations online, so Thank you very much, Anna. Alright, so our next student up, the last of this session will be Gabriel. Thank you to please stand on that side if you can manage of the screen, so we have an easier time seeing the presentation as the part of the jury. And as you can see, you can just swipe the screen.
Okay.
You want to test it before you go?
Yeah.
Yeah, you're fine. Right, super. Thank you very
Okay,
much. You have
so
15 minutes.
Good morning, I'm Gabriel Rivas and I'm going to present my master thesis. And I'll start by giving you a little introduction about the topic. So, what are animal forests? Animal forests are actually ecosystems formed by megabenthic organisms which are able to form complex three-dimensional structures and provide ecosystems to other organisms. Therefore, fisheries often target these ecosystems in the search of commercial valuable species. And at the same time, the bottom contact fishing gears of them often harm these megabenthic organisms, disrupting ecosystem services, decreasing biodiversity, and changing the benthic community. So, oops.
No, go ahead.
No, it's not. That's
Oh.
not.
Okay. Just a second. Sorry. You want to take it back from the top?
Well,
If
no,
it's
I
you-
prefer to.
You continue from there?
Yeah.
Okay. That's good. We're not too far in, we can also start. Well, we have a delay, but Tim is still in the room. There's a delay between the lights. That is good. Nah, it should come back on, yeah. And whenever you're ready, Kevin.
Okay. So one of the most damaged species are, or animals are gorgonians, as you can see here, which often get removed the gorgoning under skeletons, causing the overgrowth of epibionts and the necrosis of them. So right now the European Union and other institutions are trying to implement a series of marine protected areas or MPAs to protect these organisms. So in this context, the main aim of this master thesis is to first describe the main animal forests in the mesophotic front of the northern Catalonian continental shelf, to see if there are changes in the community composition depending on the fishing impact, and to evaluate the current conservation status of the Gorgonian assemblages in these sites. Hence, the hypothesis is that higher fishing intensity will translate into lower values of biodiversity, differences in the benthic community composition, and higher appearance of necrosis and epibionts in the Gorgonians. So let's go on how this master thesis was conducted, and I'll start by presenting the study area. So it is the mesophotic zone of the northern Catalonian coast, and we set 3 different sites. Here in the north, which each of the sites had 2 different areas, a reserve in which higher fishing have higher restrictions, and a control in which fishing have lower restrictions. So in the northern part, we can see here in red the reserve area and the control without the red. The Tancatejans and Cap de Creu sites have in the reserve, well, it wasn't allowed to do any fishing practice, while in the control, artisanal fishing is still allowed. Then in the southern part, in Voldetosa, we have, that is the only site in which in the control is still, bottom trawling is still allowed, and the reserve restricts this fishing into artisanal. We conducted a series of biotransects using remote operable vehicle or ROVs. And as you can see here, the video footage was analyzed using this software here, that is Beagle, and it is used to tag all the organisms we were able to see and identify them into the lowest operable taxonomic unit, or O2. At the same time, we categorize the different substrates into 3 categories: hard, mixed, and soft substrates. And we also quantified the presence of marine litter. After, with all this database, we analyze it in R, and what we first did is to divide each of the transects into sampling unit of 5 meter squares that acted as replicates between them, and then we were able to characterize the substrate and area composition of each of the sites with this analysis conducted here. And then we also quantify the presence of necrosis and epibionts using Gorgonian assemblages and comparing them between the different areas and building an occupancy model to estimate the total presence of these damages. So the results were that in total we were able to survey a total area of 2,745 meter squares of the Northern Catalonian continental shelf, which corresponded to 11 hours 47 minutes. And most of this area corresponding to substrate and control. Also, we encountered over 19,800 species, most of them belonging to 112 different autus, of which most of them were able to identify till the species and genus level. And as you can see here in the graph, most of the identified organisms were Cnidarian and Polyptera, and in the reserve area we had reported higher species densities than in the control. When we characterized each of the sites, first we started by analyzing the different substrate composition between areas, and the only one presented no significant difference was Tancat de Llançà, while Cap de Creus and Bord de Tossa did present significant differences. After, we analyzed the biodiversity using Shannon test. As you can see here, there were, for all of them, significant differences were reported, but in the case of Tancat de Llançà and Bord de Tossa, hard substrate and mixed substrate were significantly higher, while in Cap de Creus, mixed and soft substrate were higher. And then, when we compared the control and compare, no significant differences in the channel were found. Then also the hill numbers presented higher values of biodiversity for the control in Tancat de Llançà, similar values for Cap de Creus, and lower values in the control for Bord de Tossa. And when we analyze the community composition between different substrates, we can see here in the MDS different clusters depending on the substrate, and in fact the Permanova did show significant differences. Similarly, when we compare the community depending on the area, some unclear pattern we could find, but the Permanova at the end did show significant differences for all of the sites. Again,
It's writing.
It's writing.
Very good. Very good.
So finally, the involves it tells us the most representative species for each of the groups analyzed. But I will talk about them more in the discussion. So when we analyze the Gorgonian assemblages, in total we found 1,933 individuals belonging to 9 different species, but most of the individuals were Amygdala cingularis and Amygdala cavolini. And when we analyzed the occurrence of necrosis and epibionts in the different sites, it reported values of 24.8% of the Gorgonians in Tancat de Llançà, 25.2% of the Gorgonians in Cap de Creus, and 16.3% of the Gorgonians in Bord de Tossa presented necrosis or epibionts. But nevertheless, the only one showing significant difference between the areas was Cap de Creus, which also presented significant differences in the epibion community composition, being Archeonic coralloides in blue higher in the reserve than in the control. And finally, we built an occupancy model using depth and substrate as site cover rates and the size of the artisanal fleet and the presence of marine litter as cover rates. This legit model was able to estimate that 30.8% of the total Gorgonian assemblages will present or epibionts or necrosis. So now, what does this actually mean? So in total, we were able to describe a different animal forest in the northern part of the Catalonian coast, depending on the benthic community composition, the substrate, and depth. So in the shallower part of the mesophilic flow, we first identify an animal forest for soft substrate formed by sabellids and Altinum palmatum. As you can see here in the image, it's quite flat, and actually reporting the lower values in biodiversity because they are not able to form, these species are not able to form well-developed structures, and hence it doesn't support much biodiversity. At the same time, most of these species are short life history organisms because of the high fishing intensity. In comparison, the other animal forest reports, which were hard substrate with Unicella singularis, Paratoanthus coccinellale, sponges and bryozoans, have a higher spatial heterogeneity, providing more niches to other species. Similarly, in the deeper part, we found for soft substrate animal forest formed by soft corals and Lithocarpium myriophyllum. We cannot see here Lithocarpium myriophyllum, but here the Pernatulaceans and also the sea pens can provide these structures. Also, transforming these flat bottoms into intricate of structures. And similar in mixed substrate, we had Unicella cavallini with sponges. In this case, Unicella cavallini replaces every other Gorgonian species after 80 meters, and is, in the case of the hard substrate, which reported lower values of biodiversity, is because these species produce a series of allelochemicals and feed on the larvae of other benthic organisms, which creating a higher dominance of it and lowering this biodiversity. Then, after when we analyze the fishing impact, depending on the fishing impact, we found that the two main factors driving this biodiversity, this fishing intensity and changing the benthic community composition, were the fishing intensity and the amount of time the reserve area, which is an MPA, has been close to fisheries. For example, in Borde Tosa, which is the only one allowing bottom trawling, the main organisms in there were short life history trace organisms, while in the reserve area, which is the oldest one, implemented in 2011, have already replaced these organisms to long life history trace ones. Similar happened between the Cap de Creus and the Tancat de Llança site, which the artisanal fishing is still appreciable in these changes. But now you might be wondering, how do we know that these changes are actually negative and not positive? Well, but it's because of these four species here, which reported to be the most representative for the reserve areas, and all of them have already been reported in other studies to be removed and to have negative relationship towards fishing. Then, when we analyzed the Gorgonian assemblages, we at the end estimated that that insufficient time had passed to observe any recovery, since most of them may take from decades to centuries to recover. But as in the Tancat de Llança, it didn't show significant differences. But also in Cap de Creu, it did show significant differences, but the higher presence of Altinum coralloides, which is a parasite of Gorgonians, did show that the time wasn't enough. Also, the occupancy model was built using fishing intensity and marine litter, which have been reported to be the main drivers of epibionts and necrosis in these waters. And then I wanted to have a special mention to the co-management of these MPAs, because all the reserve areas were actually MPAs implemented as the result of the collaboration between fishers and the scientists. And now we could see that fishing intensity does change benthic communities, and the involvement of their knowledge and their collaboration is crucial to have a successful protection of these communities. So, in conclusion, we were able to estimate a total of 19,000 organisms belonging to 112 different OTUs, which serve to identify identified 4 different animal forests described. Also, we were able to appreciate that changes in benthic community in fishing density change benthic community composition. But the time passed since the implementation of these MPAs has been insufficient to observe changes in animal forest and Gorgonian assemblies. Finally, we were able to estimate that 30.8% of the epibionts of the Gorgonians will present necrosis or epibionts, but we can actually lower these values if we take into consideration the fishing intensity and the presence of marine litter. So, thanks for your attention. Any questions?
Thank you, Gabriel. You made it to about 30 seconds of the allotted time. You were speaking a bit fast at times. I think you're maybe a bit stressed. I don't know.
No, it's not because it was too much information. I knew I have to run.
No, you didn't have to run so much, but it was a great presentation. Thank you. So, whoever wants to go first?
Well, I can go first if you want.
I don't mind.
Thank you for your presentation. I have some questions about fishing. It's not really clear to me what kind of fishing we're talking about.
Okay, so in this area, but basically in the northern Qatari continental shelf, bottom trolling is allowed in all the coasts except for areas below 50 meters and some MPAs.
Okay.
In these cases, bottom trolling is only observable in the Pol de Tosa, but Tangat de Jansa and Cap de Creus were inside MPAs or shallow in areas shallower than 50 meters. So artisanal fishing is the only one who can be practiced there. But at the same time, the MPA implemented in those areas reduce that artisanal fishing to no fishing practice.
Yeah.
So I don't know if that answers your question.
Yeah, no, but concretely, like when you say artisanal fishing, what kind of fishing are we doing?
Long lines, trommel. We didn't take into consideration, for example, pursuit because only those using bottom contact fishing gears because are the one affecting the benthic resistance.
Okay, and for instance, if a troll net arrives on the Gorgonian fields, there's basically nothing left behind, right?
Yeah, most of the time, yeah.
So my question is about what mechanisms underlie this link between fishing intensity and the presence of necrosis and epibiosis.
Yeah, I mean, in these cases, most of the, in the areas we studied, bottom trolling doesn't affect the Gorgonian species. Why? First, because in the Pol de Tosa site, only Gorgonians were found in the reserve area, so no bottom trolling. And in Tancat de Jansa and Cap de Creus, bottom trolling is not allowed. So the one producing necrosis are mostly long lines, trommel nets, and that's why in the occupancy model we only use artisanal fishing and not bottom trolling.
Okay, I missed this subtlety. Why did you use Shannon index to compare your different assemblages?
Yeah, actually I wanted to, I mean it's mostly a problem of statistics.
It's not.
No, I wanted to do Hill numbers and Shannon index, but the thing is like Hill numbers I feel like it gives you a more representative evaluation of the area because also involve the species density and the species richness and the dominance of the other species, but we were not able to, the tests that I'm using are to prove significant differences, so instead I use the Shannon index to test it. So I use the Hill numbers as a kind of trends inside these no significant differences, and Shannon to actually test if there were differences or not.
Yeah, but why not just species richness and evenness if you want to test for dominance?
Because species richness, just from the beginning, hard substrate, we know that they are they have more densities than higher densities and higher number of species than soft substrate. So that's why to trying to
Okay.
avoid this.
Okay, thank you.
Okay, thanks. It's interesting work. I would say, okay, this is a nice and valuable amount of data. Can you please go back to the slide, one of the first slides where you showed the sampling design?
In the
Yeah, okay, the one with
this one.
okay, go ahead.
The map.
No, go ahead.
What? The map?
The map, yes.
Ah, okay.
Okay. Okay, so do I understand correctly that for each of these three areas, essentially you had one area that you considered control, which was the area with the protected area, and then another area that you considered impacted. And you had one, for each of the three areas, one with both the condition.
Yeah.
Look, I'll tell you, with something designed like this, I would feel a bit uncomfortable to draw to the conclusions because in principle, if you want to conclude that the reason of the differences that you found between impacted areas and protected areas, in theory, you should have at least two impacted areas, two impacted sites.
Yeah,
So
actually.
that would be, in theory, a conceptual requirement. Because that will give you an estimate of the difference between in theory. Look, I'm not saying that what you did is valuable, is useful, but in this way, I wouldn't feel comfortable to conclude that the differences that you found are directly related to the impact of fisheries. In principle, they might be due to other reasons and other factors related to natural variability.
I mean, actually, first there was another area which I didn't have time enough to analyze, but was also to compare bottom trawling and artisanal fishing in the same way as all the TOSA. But then, in general, for the Mediterranean Sea, most of the studies report that the main factor causing the damages in mostly megabenthic organisms is fishing intensity. So yeah, of course, there is a bit of lack of experimental design in this case, but is kind of aligning with what other studies have already reported.
I believe that I guess that in the practice, what you found most probably will be indeed related to fishing impacts, but from a logical, conceptual point of view, this is the type of logic which is applying in the so-called beyond bashing the sampling designs in the period of the experimental designs, which are considered the most reliable when you want to assess the effects of a human impact. Still, I'm not saying that what you did is useless. What I recommend is just to be a bit careful with the interpretation of your results.
Okay, thank you.
And actually, just a matter of curiosity, of the taxa that you consider only animal taxa or other also?
Only animal taxa.
Only animal.
And most of them megabenthic, but we also take into consideration the fish that were around the animal forest.
Okay, fine. Okay, thank you.
Any questions from the room? I won't have any questions. I would just first of all thank you very much, Javier. We are going to take a half an hour break. We start again at 11. Please, two things. You see some rooms finish before others, so when you exit the room, don't start talking in the corridor because you might disturb the next room, please. And also just try to be on time as you did this time also. So, and pass the word around for the rooms, because the other ones finished early and they did not have that problem, alright?
Okay, thank you very much, Mary. First, welcome everybody to this session too. My name is Eva García Vázquez. I come from the University of Oviedo in Spain. I will be the chairperson in this session, and very briefly, you have 15 minutes for your presentation, then another 15 minutes for questions from the different evaluators, and if I have from the evaluators, here Margarida Castro from the University of Algarve, and Gauthier Jacques from the UO. If there is room for one more question, I will pose also some questions, okay? And if there is still room, the audience can pose questions as well, okay? So, the first presenter in this session is Brigitta Varga, and she will speak about the marine megaherbivore Thesis enhanced seagrass seed germination. So the floor is yours. You have 15 minutes.
Good morning, everyone.
Excuse me, Brigitte, can you please sit on this side of the screen, and you can switch the slides just by swiping on the screen.
Yeah. Okay.
Yeah.
So like that you're not between us and the screen.
Good morning, everyone. My name is Brigitta, and I'm going to introduce you my thesis project. I carried out this project at James Cook University at the Trollwater team in Australia, under the supervision of Dr. Samantha Toll and Dr. Robert Coles. When I'm talking about marine megaherbivores, I'm going to refer to green sea turtles and dugongs. Green sea turtles are currently listed as endangered, and the dugongs are listed as vulnerable by the IUCN due to their population decline. Seagrass is a crucial component of this species diet, and the increasing decline of the seagrass meadows, their survival is threatened. Seagrass is not only a vital food for marine megaherbivores, but also provide habitat for many fish, bird, and invertebrate species, and they also play an important role in the ocean's carbon, phosphorus, and nitrogen cycles. To encounter these losses in the seagrass meadows, restoration efforts are increasing worldwide. In Australia and New Zealand, the seagrass restoration projects have focused on small-scale experiments through planting seagrass fragments and/or seed-based restoration efforts. The fragment-based restoration is more expensive and takes more time, but it has a higher success rate. But the seed-based restoration is cheaper, doesn't require a larger team, but it has a low success rate, mostly due to the low rate of the germinated seeds. Sea turtles and dugongs are known as seagrass seed dispersers by consuming and excreting the viable seeds into their feces. And the study found that these excreted seeds have a higher germination probability, and they germinate faster than the seeds dropped by the plants. And there are some possible mechanisms behind this, like the physical scarification of the seed coat, sterilization from fungal or bacterial infections, or an increase in available nutrients. The aim of this study was to investigate if the marine megaherbivore Thesis increase the available nitrogen and phosphorus for the seagrass growth, if this increase enhance the seed germination and the seedling survival. And this study could have a positive effect on future seagrass restoration efforts and ultimately benefit green sea turtle and dugong conservation. This study focused on Zostera muelleri, the most common found seagrass species in marine megaherbivore Thesis in northeastern Australia. And here you can see our collection sites. The seagrass seeds were collected from an intertidal seagrass meadow in Gladstone, and the Thesis samples and the sediment core samples were collected from two mixed species seagrass meadows in Townsville. The seeds were collected by the Central Queensland University in December, and they were kept in dormancy until we received it. And we collected the Thesis samples from both during high tide. They were floating on the surface, so we could use a net for that. And the sediment cores were collected during low tide. We used the PVC pipes, and we kept only the top 5 centimeters of the cores because that's where most of the seeds can be found. So the seeds were randomly allocated to either feces or non-feces treatment. The feces treatment seeds were kept in a nutrient bath for 7 days because that's the length of half of the time of sea turtle digestion and the full time of dugongs. And so 2 feces from the February collection were divided into 30 vials with a hole in the lid and the mesh cover to keep to allow water flow, and 147 seeds were divided into the vials, and then they were left in this small aquarium. We maintained the salinity at 35 ppt and the temperature at 28 Celsius degrees, and we also had a bubbler, and we covered the aquarium with an aluminium foil and a tray on top to keep darkness, as it would be in the digestion tract of a marine megaherbivore. And so the seeds were placed in a flow-through aquarium system. We had 8 Nelly tanks, but we ended up using only 6 for this experiment because I ran another one in the other 2. And so we had 3 tanks per treatment, and we had 6 small plastic containers in each tank, and then we had 3 seeds in sterilized autoclaved sand buried. So we had 54 seeds per treatment, and then all the statistical analyses were conducted in the R program. Because the response variable was left skewed and the transformations were not successful, I ended up running a mixed effects multinomial logistic regression model. And with that, I was able to analyze the difference in seed germination based on treatment. The response variable was the outcome that categorized the seeds into 3 groups: the dead, the viable, and the germinated. And the treatment was included as fixed effect with 2 levels. Tank and replicate were included as random effects, and VRMS package were used because of this, because its flexibility with the non-distributional data. And I also tested the viability of the seeds by tetrazolium staining. So for that, I removed the seed coats under a microscope, and then I left them for 48 hours in darkness with the tetrazolium dissolution. And here you can see the non-viable seeds, and here the colorization in the cotyledon of the seed. So this is the viable one. So the starting percentage of the viable seed for the non-feces treatment was 48%, and then this reduced after the nutrient bath to 24%. The storage time usually is a maximum of 7 months for the Zostera muelleri seed, with up to 58%, but our research shows that it can be much less, because here we had 3 months of storage and it reduced to less than 50 already. So we recommend the use of the fresh seeds for the experiments whenever it's possible. And the viability loss after the nutrient bath could be due to the rapid addition of the high nutrients, which could be resolved the diluted version of the nutrient bath or with a reduce time for the nutrient loss. And after by day 63 of the experiment, there was no sign of seedling, so I sieved all the sand and retrieved all the seeds. And then for the non-feces treatment, we got 92.6% dead seeds, none of them were germinated, and 7.4 were ungerminated. So I did the staining, and 75% were viable. And for the feces treatment, we had 72.2% dead, and 7.4 germinated, 20.4 not germinated, but all of them were viable. So the marine megaherbivore feces significantly increased the number of germinated seeds, which suggests the potential use for seagrass seed germination project. Here we can see on the x-axis the outcomes, the dead, the germinated, and the viable outcomes. On the epsilon axis, the number of seeds per treatment, per replicate, sorry. And the brighter blue shows the non-feces results, and on the right, the feces results. Individual counts the outliers, and so we have an overall quite low mortality and small amount of germinated seeds. We sent the feces and sediment core samples to another laboratory, to Townsville, to analyze the concentration of nitrogen and phosphorus, and we got the result that the nitrogen levels were 135 times more in the faces compared to the sediment, and the phosphorus were 30 times more in the faces compared to the sediment. And we did this to see if the higher germination was due to the higher available nutrient in the feces. And so we can also see some difference in moles. So the nitrogen decreased and the phosphorus the opposite, it increased, and both of them could discuss by the senescence season of the seagrass. The nitrogen, this time the nitrogen are being absorbed, so that's why it could be reduced in the concentration. And the phosphorus is less mobile in the plant, so it tends to accumulate, so that could be the reason why we found a higher amount of that. And to conclude, this study show insights into the potential use of marine megaherbivore faces to enhance seagrass seed germination. Also, our findings suggest that the feces from dugongs and green sea turtles increase the available nitrogen and phosphorus for seagrass germination. The high number of ungerminated but viable seeds indicates that there might be a delayed effect of the feces in the germination, which could be resolved by additional time for the experiment or other optimal conditions for successful germination. And moreover, the high overall mortality rate and the low germination rate could indicates that we should further investigate into factors like storage duration or salinity variation or gradient nutrient addition. And our findings are promising for future seagrass restoration efforts and green sea turtle and dugong conservation. Thank you.
Thank you very much, Margarita. So now we go to the questions. Margarita, please.
Thank you. Nice presentation. I have a few questions that have to do with the connection between your experiment and reality. And I'm going to ask you two questions. I just want to know your opinion,
Yeah, sure.
okay? First one is, you simulated the digestion of the seeds inside the gut of a turtle.
Only the time and the darkness, not the acids.
Because, you know, if you have seed scarification as being part of this, the digestion is very important for increasing the viability of the germination of the seed. So maybe you have a low value for the germination. Maybe you get better results with a natural digestion by a sea turtle. I don't know if these seeds need that or not, but many seeds need digestion to be able to be more viable. The other question has to do with the concentration of the feces. You use basically pure feces, but in the natural environment, the feces will be diluted and spread out, and the concentration of seed of the feces mixed in the sediment will be much lower than the one we used. Am I correct?
So the feces is floating on the surface like as a whole, so it's not really diluted.
Yeah, but when it gets to the sediment, if it's going to fertilize seed germination, eventually the nutrients have to be moved to the soil, no? To the sediment.
Okay.
And do they go in the same concentration as a full piece of feces?
Yeah, that's a good question. Yeah.
My question is, shouldn't you have an intermediate level where you mix feces with sediment, but not pure feces, or not with a lot of feces? See, in the design of the experiment, to have no feces, some feces, and a lot of feces, to see, you see, another level of your treatment, to be able to have a more realistic situation.
Yes.
Maybe a suggestion
Yeah,
for
this
future
would
experiments.
be a first try, let's say.
Okay.
Yeah, but so after the nutrient buff, we planted them to the sediment as well. So.
Well, the last question is, you showed in your methodologies, you applied mixed models, regression mixed models. You didn't show the results of those models.
Yeah, because I mean.
You're still going to.
Yeah, so I didn't put the exact parameters, but because they contained 0, they were significant because they don't give the p-values, so I only had the.
Because you had 0 and you could not do the.
Yes, so they were
okay.
significant, and that's why I didn't include
Okay.
them.
All right, thank you.
Thank you, my friend.
Thank you. Yeah, thanks for your presentation. And yeah, I think most of my questions were also about how the results from your experiment can be transferred into the natural environment and be insightful. And so maybe I can start with that. When you in your last slide you say that your study can be interesting in a seagrass conservation perspective. So can you develop on that, please?
So because the germination rates are quite low, and there are quite a small amount of study in this to improve these rates, I think if they try to implement our techniques, this could help to do restorations by first doing the faces treatment and then doing the restoration.
So okay, so you say you should basically incubate your seeds with dugong and turtle poo.
Yes.
And then okay, does it has to be dugong or turtle feces?
Yeah.
I mean, if they just need nutrients, I'm pretty sure there are some cheaper sources of nutrients.
Yeah, we also added some nutrients to the water.
Yeah.
I didn't mention that, but so the boat treatment has an average level of nutrients according to the level in the coral sea. So we had a normal amount of nutrients.
Yeah. But probably incubating your seeds with cow poo would be exactly the same and much cheaper. No?
I don't know if it's cheaper, but yeah.
Okay, and you also say it can be interesting for turtle and dugong conservation.
Because seagrass is their main food source, and
Okay,
because
that's a
of the
consequence
decline,
of seagrass.
yeah.
Okay.
Yes.
Okay. How many turtles and dugongs there are, like in the bay around Townsville? Did you try? I mean, even with rough estimates, did you try to estimate how many individuals there are, like, and how many feces they produce, and what is the concentration of feces in the water and compare it with what you did, where I saw like an aquarium, like covered in feces in vials. It was a very interesting experiment.
Yeah, so I'm not sure about the exact numbers, but when I was there for 3 times on the boat, I've seen at least 10 sea turtle at that time and 3 dugongs at the first time, and none of them by the end, because we also observed that there was almost no seagrass left because we had a really hard cyclone and flood, so there was barely any seagrass, so they probably just went somewhere else because there was no food for them. So yeah, and so the poo, I collected everything I found, so it wasn't that much.
So like the water is not covered in floating feces?
No.
Okay.
No.
I have another question. Never talked that much about poop since I was 5 years old. Why did you use some sterilized autoclaved sand for your experiments?
Because we didn't want any other condition to have an impact on our experiment.
Yeah, but don't you think all the microorganisms in the sand can have an effect on the germination of
It can,
seeds?
but we wanted to know if the nitrogen is the one. Nitrogen and phosphorus is the thing that's helping them to germinate.
I understand, but I was wondering because I know that fungi, for instance, can be important in helping with germination, and I mean
Yeah,
the positive interactions between the rhizosphere and the plants are quite important, and they can maybe, I'm not a specialist at all, but can also contribute explaining the such low germination rates.
Yeah, absolutely. That was one of the possibilities, but we chose to try the nutrient one.
Yeah. Last, it's a very minor comment, but can you go back to this box plot, the last box plot you showed? Yeah, this one. How many data points did you have for each condition? Yeah.
So I had 54 seeds per treatment.
Yeah, but I mean, it seems here that your box plots were built with only 2 or 3 points of, and then they can generate the quarters with even 2.
For this one I used only, I counted it as how many out of the 3 that we have per replicate was that germinated or what?
Yeah, but I mean, is it 3 or 4 points? But basically, when you make a box plot, you need at least 5 points, because there has to be a median, the quartiles, and the extrema. And it seems here, from what I see with the yes conditions, that you don't have those 5 points because you don't have the whiskers on the box plot. Yeah. So you probably don't have enough points to make a box plot. That's just a detail, but I think it's important for everyone to realize that representing data with a box plot requires at least 5 data points. Okay, thank you, Anita.
Thank you, Gauthier. So, I think we are behind time, so we have like 6 minutes left for more questions. So I will make you a few observations and then a few questions. First, congratulations for the work. I see the implications of this study for conservation, both the macrophytes and the species that are pulling on them, more or less. So I think your work has lots of implications and also perhaps some applications. But you may develop on the applications later, okay? First, my observation is about, I missed somehow the references in the introduction and in the discussion, some references to compare your results with other people.
Yes.
So from what you read, I'm sure you have prepared a lot of this work, so I'm sure you read a lot of articles. What did you expect when you started the experiments? Did you expect the thesis, the pool, has some effect on the germination or viability, or
Yeah.
is it totally novel, this study?
And this is the first time I think someone ever tried it.
You
Yeah,
should
but
tell this at the beginning. This is super novel. To sell the product.
yeah, so
What did you expect?
I expected to have a much higher germination rate, obviously, but we had some significancy. So
Yes.
I'm happy with that. And if I could do again, I would extend the time to wait until they seedling, but I didn't have time to wait for them. So yeah, so I definitely expected higher germination rate. But yeah, and first we wanted to use another species which germinates faster, but we couldn't find any due to the flood, so I had to use these, and they germinate quite slow. Sorry.
So perhaps it was too short time
Yeah, definitely.
for this
Yes.
species,
Yes.
for the plant.
And also because we received these seeds after storage, and it seems like they don't like to be stored, or at least not in these conditions.
I liked very much one of the questions about the number of dugongs that are around and the number of sea turtles compared to cows, for example, that are adding food to the environment. But well, I had a similar curiosity about this. So how important as components of the community are these two species, and why did you choose these two species in particular?
I chose them because they are present in that area. These areas are known as foraging areas for both of them, and also because my supervisor did not a similar, but she did research on the feces, the feces, and she was retrieving the seeds from the feces. And because they had higher germination, and I was interested in why was it, because they didn't know it before.
And these are the natural herbivores, so they feed on this plant, the two species.
Yes.
So this is the main reason.
Yes, that's main. Yeah.
This is good. It's not like it's not as in reality, but at least it's connected with reality. So, if you did the experiment again, what would you change besides the time, waiting
Yes.
time for the
Yes.
germination? But,
Yes, so I would use the other species first. And also I would try other salinities, because, for example, for this species it's known that if we add freshwater pools, they germinate faster, so I would try that one as well. Yeah, and I would try to find the best way to do the nutrient bath. Maybe it was too much time in the bath because of the high mortality rate, so I would try to change that.
or perhaps to use intermediate concentration.
Yes.
This could be another possibility, because I assume that the density of the herbivore populations changes over time, right?
Yes.
It's seasonal, or are they present all time?
As far as I know, they are present.
Mhm.
They are
So they are resident there.
Yes, I would say yes.
And about the relative abundance of species of turtles and dugongs, could you roughly estimate what are more abundant in situ, in play? I mean, there are more pools of the turtle or
Oh yeah, so we couldn't identify the pool, that's why we call them marine megaherbivores, because without a genetical analysis, that is not possible to differentiate the two. So, and there was no equipment to do this analysis at the level.
I guess they are not floating forever. I suppose they are
Yeah,
dissolved.
after a while they are frozen.
They have remains. Okay, thank you. There is time for one question from the audience. Yeah.
I just had a quick question. It's more like curiosity. I don't expect you to have the answer. But, I come from Brittany, and we have a lot of issue with green algae,
Yeah.
and because we have a lot of nutrients in the water. So just did you consider the fact that too much nutrients could actually be harmful for the seabeds?
Yeah, so yeah, we already got the results that the high nutrients is not the best, so that's why I would try to find the best concentration of
Yeah.
nutrients. Yeah.
But my question was, I will try to.
Sorry, I need to stop the discussion.
Okay.
Okay.
You may discuss later because these are very interesting results.
Okay.
Okay,
Thank you so much.
very much.
Thank you.
Very enthusiastic.
Next presenter is Sofia Ulkov. And her work is Fishing and Seafloor Sustainability towards an integrative approach for assessing bottom trawling sensitivity in the southern North Sea. So, the floor is yours.
So, hello, my name is Sophie, and my master's thesis was on the topic of fishing and seafloor sustainability, and more precisely working towards an integrative approach for assessing the effect of bottom trawl fishing on seabed sensitivity. I did this work with the Flemish Agriculture and Fisheries Research Centre, and my supervisors were Gert van Hooy, Joanna de Smet, and Jochen Deppestelle. I picked this research topic because I have a personal interest in bridging the science policy gap, and I wanted to gain a more in-depth understanding of the mechanisms and metrics that are currently used to inform the effect of bottom trawling on the seabed, which, as I'm sure most of you know, is having an increasingly ecological demand on the seabed, and the correct and robust development of indicators for measuring the effect is pivotal for informing and preserving marine health. So what is an ecological indicator? Well, these are tools that combine multiple variables and factors into one outcome. These, the complexity and variation in the measurement methods have resulted in a very large array of indicators, which sometimes have contrasting results and therefore confusing messages for management. The lack of standardization, well, these are measured on biogeographical, regional and national scales, and there's a lack of standardization currently between and even within regions, which produces very confusing messages for management. In this project, I looked at 5 indicators for bottom trawling. The FBIT indicator, which is developed by the ICES working group for the fisheries benthic and impact trade-offs, is what it stands for. And it looks at the median longevity of species within an area to determine the sensitivity of that area to fishing. I also looked at the BH3 indicator, which is looks at the resistance and resilience of an ecosystem to bottom trawling. So it's more of a risk-based indicator, looking at the impact of fishing. I then looked at 2 biological trait indicators, so the benthic sensitivity index to trawling operations, otherwise known as BSITO, and the trawling disturbance index, otherwise known as TDI. These use biological traits of infaunal species or epifaunal species, depending on the study, to determine an overall score for the sensitivity of that habitat. This can be done on infauna or epifauna. Then I also looked at a diversity index, so the Markov diversity index that compares diversity from an undisturbed state and a disturbed state. And it has been adapted for measuring fishing pressure in particular. So what am I going to do with these indicators? Well, first of all, I'm going to see whether the indicator outcomes are comparable, whether for the same area they are saying that the area is sensitive or not. What are the factors that might be influencing the difference in indicator outcome, should there be one? And are the indicators responsive to trawling pressure, the things that they were made to indicate? What are the trade-offs involved in integrating these indicators into one overall value? So that is taking the indicators that are all calculated from different variables and then integrating them into one outcome to have a more comprehensive and clear value to be able to communicate the sensitivity to managers and policymakers. I then wanted to create a heat map, such as this, of the TDI, Maglev, diversity, and Mesito outcomes to determine whether spatially they display the same areas as being sensitive or not. So in a nutshell, before I go into the methods and results, I found that the indicator outcomes were for the most part not comparable, which warrants caution in using one single indicator to look at the sensitivity of an area. Habitat classification influenced indicator outcome. Mesito and TDI indicators showed a lack of responsiveness to trawling pressure. And a heat map of indicator outcomes displayed the same areas as being sensitive to trawling pressure, providing some evidence that spatial integration of indicators could provide a more complete overview for seabed sensitivity, for application in fisheries management and providing a visual tool to aid this. So what ecological data did I use to get these results? Well, I used data from Belgium, the Netherlands, and the United Kingdom, from 2 fisheries management organizations, and filtered the data between 2010 and 2021. I only used infauna records for this analysis, and I isolated the dataset for the Southern North Sea region. I then added environmental data and fisheries data to the final dataset. This was the distribution of the samples. The dark colored points are the records gained from the CFAS UK organization, and the yellow points were gained from the Belgium and Dutch Institute. So first and foremost, I did a preliminary analysis to assess whether there was an influence of habitat classification on the 2 indicators FBIT and BH3. So not all 5, just 2. The 2 habitat classifications that I looked into and calculated the indicators with was the MSFD habitat classification, which is used to inform the Marine Strategic Framework Directive on the health of the marine environment. That is the EU legislation. I then also used the UNIS habitat classification, which is a more distinct and fine resolution habitat classification that takes into account finer detailed physical, chemical, and biotic variables. And this is what I found. So one would expect that with the difference, each habitat is very different. You have rocky habitat, you have muddy habitat, you have sandy habitat, and the indicators these habitats are in theory very different to their sensitivity to bottom trawling. So we would expect there to be a large difference in mean sensitivity of the FBIT in red and the BH3 in blue and yellow to bottom trawling. These are the 5 largest habitats in kilometers squared for each one of the habitat classifications. What we can see is that the MSFD habitat has there's much less variation in mean sensitivity
in two indicator outcomes for this habitat classification as opposed to the UNIS one, which has more difference. From this we conclude that habitat classification does indeed have an influence on indicator outcome, and that the UNIS habitat classification with its finer resolution is more appropriate for assessments of benthic health. Secondly, I did an indicator comparison. First, I did a correlation analysis of the Besito, TDI, and Margolov diversity indicators on both the sample level and UNIS habitat 4 level. The two biological trait indicators, so the ones that were in yellow on the previous slide, the TDI and Besito indicators, came out to be less correlated than TDI and the diversity indicator, Margolov. This is a surprising finding given their similar method of calculation. However, when looking at the ranked mean sensitivities, again for the 5 largest UNIS habitat types, we can see with 1 being the least sensitive and 5 being the most sensitive, we can see that this relationship is different as TDI and Besito come out with identical rankings, whereas for the other 3 indicators, there's much difference in ranking. So from this correlation analysis, we can see that the scale to which we calculate indicators really influences the way we interpret the sensitivity of seabed habitat. Thirdly, I evaluated whether the indicators were actually indicating well for bottom trawling. I used fishing data from OSPAR and clipped it to the southern North Sea, and this was my outcome. So here we can see the Besito outcomes on the x-axis, so from 2 to 5, and increasing fishing effort on the y-axis, and increasing fishing effort on the x-axis, from negligible, very low, low, medium, high, and very high. There's little variation in the Besito outcomes with increasing fishing effort, which is surprising given what we would expect to see would be something like this. A stepwise increase of Besito sensitivity with increasing levels of fishing effort. This trend remained the same for the other biological trait indicator, TDI, that is calculated in a very similar methodology. The Margolov diversity indicator showed more responsiveness to trawling with a decrease in diversity with increasing fishing effort levels. I did the same analysis on a habitat level, again for the 5 largest UNIS habitat types. And although the same trend remains, so little variation of the indicator outcomes with increasing levels of fishing pressure, each habitat has quite a distinct responsiveness to trawling, which does beg the question as to whether perhaps a more precise integration of indicators should be done on a habitat level as opposed to an area-wide level. Lastly, I created a heat map of TDI, Margolov diversity, and Besito outcomes to assess whether spatial integration could be used in fisheries management, providing a visual tool to depict what areas were sensitive and less sensitive, and guide fishers towards the least sensitive areas. And these are the results. So in all indicators displayed the northeast of the area studied and the UK as being highly sensitive, so in yellow. They also displayed the eastern coast as being least sensitive with high areas of blue. The Dutch and Belgian coastlines, however, did not come out as being highly sensitive, which is an interesting outcome given that this is a very intensely utilized area for both fishing but also many other pressures. To see whether we can spatially integrate these indicators, I plotted the indicator outcomes on a map, and I took the mean sensitivity per 9.6 kilometers squared grid cell.
Sorry, Sophie, 2 minutes left.
Okay. squared grid cell to see whether they highlighted the same areas still in comparison with the other individual maps. And we can see that indeed it did, but there is a loss in sensitivity across the gradient. So there's a loss of precision of the indicators that is in the green in between yellow and blue. So to conclude, contrasting indicator outcomes do warrant a cause for concern when using one single indicator to assess the sensitivity of a seabed. We can see that perhaps Yunus habitat classification, well, in this case, Yunus habitat classification was better suited for benthic health assessments in this region again, because this is again not applicable to all the regions. And that habitat classification does influence indicator outcome. The lack of responsiveness to trawling of Visito and TDI prompts further questioning as to whether this was a true lack of responsiveness to trawling pressure, or whether it was indicating that the community has reached an adapted state to chronic trawling pressure, and perhaps further development of other indicators or investigation into this, looking further into the community, to assess whether these indicators are still being responsible to trawling. Heat maps and spatial integration of indicators could provide a more complete and accessible overview of seabed sensitivity to be applied more immediately in fisheries management without the confusion of many numbers and many abbreviations, and developing a more clear and comprehensive view of seabed sensitivity. So the next steps in this project formed the basis of a larger project with the Fisheries Institute that aims to develop just that, a seabed sensitivity map for the Belgian fleet to use in their operations as an onboard tool. More indicators need to be compared for this, and the influence of environmental outcomes also. Further development of a spatial integration method would be very beneficial in developing a clear, appropriate map for better fisheries management, taking into account seabed sensitivity in the North Sea and beyond. And that's it.
Okay, thank you very much. Now the questions from the evaluators. First, Gauthier.
Maria ordered me to start, so I comply.
Okay.
Thank you very much, Sophie. It's a super interesting work. It's quite hard to follow for us if we're not familiar with all those indicators. It's very complicated, but I'm deeply convinced that those studies are really necessary in a time where we see many indicators flourishing here and there, and in the end, the link between science and policy that you mentioned is made really difficult by the evidence of these indices. So I have several questions, and I apologize ahead because I may have missed something that you heard actually say. The first one is, why did you choose to focus on InfoNA?
This is a very interesting question, because when taking into account the longevity studies, so within the indicator calculations, longevity is a parameter, and there's a lack of information with regard to that, which would make, if we took InfoNA into account also within these studies, the results would be quite inaccurate given a lack of sufficient base information as an input into the indicator calculations.
But what do you think about the relevance of basing your bottom trawling effects assessment on potentially the compartment that is the least affected by bottom trawling?
Well,
not saying it's not affected.
Exactly, that's the conception, is that it's least affected by bottom trawling. But given the results of the Visito and TDI outcomes, it shows that this community, that it's not necessarily adapted to bottom trawling in any way, but there's in fauna is worms, it's critters, it's things that are not perceived as being economically valuable or viable. Therefore, focusing a study on that was of interest to me because I think it's important to communicate the fundamental basis of many of the higher ecosystems that depend on that. Valuable fish like sole, plaice, shrimp too, they all depend on infauna as well. Not solely, but a large part as their diet. So if you are altering and affecting the infauna, you are by default altering and affecting the epifauna and higher, more valuable levels.
Yeah, I agree with that. But there's also a risk of not seeing any effects on the infauna, while there might be a very strong effect on other compartments, and then there's a risk to miss some effects.
I'd rather be precise than study something else with imprecise.
You say when you're missing something, you're being precise. No, I understand the point. It was just a joke. What do you mean by sensitivity? First thing first.
Yeah. So that definition very much it changes according to the different indicators that you're calculating. So for and they are measured on different scales. Also, these indicators, which is also a root of this problem, is that each indicator does also have a different threshold for sensitivity.
Mm-hmm.
And in the group that I was working in, that was the main lead-on question: is actually what are the thresholds that mean that this environment is sensitive or this is being exploited to an extent that it's not sustainable, which is the definition of good environmental status under the Marine Strategic Framework Directive. So that is a definition that is indicator dependent, which I'm aware is such a cop-out of an answer to your question, but it's highly challenging to determine these thresholds given the complexity of the variables, and yeah, that's I hate that I can't give you a more precise answer to what is sensitivity, but it is.
I think it's very important, and I actually I started thinking about this question when you say that you were kind of puzzled to see that there was no correlation, no link between fishing pressure and sensitivity. Don't you think you can imagine an ecosystem that is extremely sensitive, but not affected at all by fishing pressure?
It's possible.
Let's imagine like deep sea corals, like the madrepora corals, they're extremely sensitive, yet, that doesn't have to be a relationship between sensitivity and fishing pressure. Or maybe
I think that's a very, I think it's to some extent a point to which you believe that in theory, everything is sensitive to any pressure that you put onto it.
Yeah, but you can imagine that biogenic habitats are much more sensitive to.
Sure, and that's also what is the decree under European legislation too. Biogenic habitats are deemed to be more sensitive. So you're asking whether it's a surprise?
Well, the thing that sometimes I had the feeling that we were getting confused between sensitivity and ecological state.
Yeah, but that's also because there are, so the two, if I get it up again, if I'm correctly understanding your question.
I'm not sure I am understanding the question.
That's reassuring.
If I'm being very honest.
Here. So there are two types of indicators actually that are currently used to assess seafloor health, and I decided not to explain this because I thought it would be a bit more confusing. But there are state-based indicators and risk-based indicators.
Yeah.
So the state-based indicators are BSITO and TDI, and these indicators are looking at the ecological state as it is, taking into account the biological traits that infaunal organisms have that would make them more or less sensitive to bottom trawling. For example, motility if an organism is stationary, for example, you were talking about biogenic reefs, they're not mobile, and the organisms that live in there usually reside in there, which would make them more sensitive to trawling, whereas an organism that can swim away is in theory less sensitive to trawling. So these are these are things that have been determined by expert judgment by the indicator development working groups that exist to determine what is sensitive and not sensitive. Again, flexibility, if something's flexible, it's less sensitive. If it's straight and can be broken, it's more sensitive. Whereas for example, the BH3 indicator is a risk-based indicator. So it's looking at the risk of fishing pressure to a given area and integrating the existing knowledge of fishing pressure within that cell on a map. And looking at the impact, and I'm not going to go too much more into it.
No, but that's an excellent response actually. Yeah. Thank you very much.
Thank you, Patrick and Nathalie.
Hello, Sophie. Well, very nice piece of work and rooms for lots of discussions, which is a good sign. I'm starting with the data. So your indexes were done before. I assume you worked in a team involved in fauna. That's why there's so much focus on InFauna, no?
No, not necessarily. I worked in a team that focused on in precisely this project, so indicator integration and looking into thresholds too.
But the origin of your data is previous samples that were done in this region.
Yes.
When?
From 2010 to 2021.
Okay, and the OSPAR data you got for the fishing pressure is for what years?
That's a good question. It's the latest OSPAR data, which was the most recent. I think it was from 2021.
Okay.
Yeah.
Because the fishing pressure, I assume beam trawling is the rule, no? Or you have auto trawling as well in that area?
Bottom trawling.
Bottom trawling, but what kind of gear? Beam or otter?
It's all of the bottom trawling.
Yeah, but you have a big difference between the impact of beam trawling and otter trawling.
Yeah.
I would guess that beam trawling, although they're smaller, they have a much bigger impact on the bottom than otter trawling.
Yeah.
So you don't have the distinction of both.
So in the fishing data, it's calculated, for SAR, so swept area ratio, which takes into account the
the swept area ratio.
Yeah, swept area ratio.
It being much bigger in an otter trawler has a lot less influence in fauna than the beam trawler. So, but anyway, if you don't have that information, you don't have that information.
I mean, at least you have like the dimensions of the gear type though within that calculation.
Okay.
So if the swept area ratio, it's impossible to know exactly the precision and tracks.
Yes, I understand that.
Satellite data.
That is the distance affected directly by the gear.
Sure.
But what I'm saying is that if it's beam trolling,
Okay.
the distance is smaller, but the whole area is deeply impacted. If it's auto trolling, you have a higher pressure on the door side, but in the middle, the net just goes over it and doesn't have such strong impact on the in-fauna.
Sure.
Okay, so probably the other fauna to make much
The InFauna disagrees with you.
the fauna disagrees with me? Okay, good. That's good for Sophie's work. So my question is, you're working with an area that has been trolled for thousands or almost hundreds of years.
Yes.
100 years at least. So you expect an index that is, I would expect the bottom to be stable and adapted to a very intense trolling pressure.
Yeah.
And I would like to show that graph where you have the fishing pressure and the indicators.
The box plot.
Yes. Well, it's box plots, is it? Oh, no, some are
This one?
that one.
Yeah.
None is what area of n is none, is what is n there?
Negligible.
Negligible.
So that's with fishing pressure. So all the fishing pressure is based on SAR, so the swept area ratio calculation that is used by OSPAR and others. Negligible is 0 to
Okay, but my question is, that index, higher values are stronger impacts, or better condition is the higher value or the lower value?
better condition is the higher value.
It's the higher value.
It's on this again. This is a state indicator.
Okay. So worst condition is the bottom points, and you have worse conditions in the one that have less fishing pressure. You see what I'm saying? That this does not seem to be good to express fishing pressure. No? So bad condition is low values. You're saying that this index, bad condition is low values.
So I'm saying that. Okay, maybe I should explain this better. So I think I've understood your question, but I'm not sure.
So what is good condition and bad condition in that scale?
It's not necessarily condition, because it's more, well, this is that the area, so low is that it has low sensitivity. So if you fish it, then it's going to, it's not going to be that impacted by fishing. If it is a highly sensitive area and a highly sensitive sample, then if you fish it, it is going to have a larger impact on the organisms that live on it.
Looks like some confounding on low pressure and low sensitiveness, otherwise this doesn't make sense, you see. So my question is, I don't think you're using the right tool to evaluate fishing pressure.
Mm-hmm.
And my question is, the second question, almost unrelated to this, what indicators are used in the Marine Strategic Framework used for integrity or for signs of pressure? How do those indicators compare with the ones we used? Did you look into them?
Yeah, so the ones that I used, so if I get up the other...
You use the classification of habitats, but not the indicators of the Marine Strategic Framework.
Are very much dependent on the area that is evaluated. So the indicators that I looked at, what are they? The indicators that I looked at, so the FFIT and BH3 and Maglub diversity, are used by OSPAR, so the Oslo and Paris Convention for...
I know, but I'm asking about what Europe is using now as to monitor the bottoms, which is the descriptors and the indicators of the Marine Strategic Framework. We didn't look into those.
Well, it's the D6 descriptor, so all of this work falls under the D6
And they
descriptor.
use the same indicators?
They use there are many more indicators that they use. So derivatives of the TDI indicator and TDI blah blah blah. They also use functional diversity indicators. So Visito is a relatively newly developed indicator that is integrated into a functional diversity indicator that is being seen as a more promising indicator to be used for informing this. Informing the Marine Strategic Framework Directive. All of these are relevant to the temporal and spatial context because they are currently, Maglub diversity has been proved to be the best diversity indicator for the region. But this is only a very small selection of all of the indicators that are currently being explored because, as I explained before, there are so many different metrics taken into account for all of the different habitats that exist.
I understand that. So my question is, don't you think that you need different indicators for different types of bottom?
Yes.
Which is not picked up there?
Yeah, sure. Yes, so I did a habitat analysis where, if this goes a bit faster, well, first of all, I say that with this graph for differences in habitat classification. And then also, if we can get them. Yeah, so these are different kinds of bottoms, and these are the ones that have the habitat classifications that have the largest kilometer squared area within the area that I studied. And I mean, that you can see that there's a distinct response to the indicators for each habitat, which suggests.
Okay. All right. No, I'm done, Sophie, because we have to end. This could last all day. Good job.
Okay. So thank you very much. Thank you very much for the presentation and also for all your answers. It's good, a good signal that you raised a lot of questions and interest about the topic. So thank you.
Thank you.
And last but not least, Maria Galdeira, speaking about the marine protected areas in the Philippines.
What is it?
Okay.
Can be very challenging. Not just a small slap behind the head. Yeah.
Yes.
Hello everyone, my name is Maria and today I'll be presenting my master thesis project which I looked into management effectiveness of marine protected areas in southeastern Mindanao, Philippines and its impacts on climate resilience and food security of small-scale fishing communities. And this project was hosted by the Oriental State University and supervised by Professor Edison Macusi and Dr. Leah Jimenez.
Control L.
Control?
L.
Full screen. Yes.
Oh, sorry.
Full screen.
Okay. Okay. So the Philippines has a rich marine life, being a hotspot for marine biodiversity, and its communities highly depend on its marine resources, being that the fisheries is a big economic driver for the country. However, decline in fish catch has been observed due to factors such as overfishing, marine pollution, and climate change. Due to the location of the country, there is a high risk for disasters and extreme weather events such as typhoons and heavy rains, which are becoming more intense due to climate change. This, coupled with more predominant oceanographic changes, have been resulting in the decline in fish catch. And of course, that small-scale fishing communities are extremely exposed to this impact, as most of the times the fisheries is the only source of income for the household. And this sector is also, it also holds one of the highest poverty indexes in the country and are extremely food insecure. This is an example of the boat they use, and it holds up to 3 people sometimes, so it's really small. However, marine protected areas can serve as a solution because if they are well managed, they can bring both social and ecological benefits to the area, increasing climate resilience and sometimes having spillover effects to its adjacent waters, which can in turn increase food security for the fishers in the area. Unfortunately, the MPAs in the Philippines are known to be under-regulated and overfished, and for this reason, we decided to look into 4 different MPAs in the area to try and answer these 3 questions. So first, is the management of MPAs in the Philippines effective? Are the communities near these MPAs resilient to climate change and food secure? And does the management effectiveness of MPAs affect the climate resilience and food security of these communities? So this is a map of the area. So like I said, we looked into 4 different MPAs, one located in each municipality. So here we have Mabini, Lupon, San Isidro, and Mati. And here in a close-up we have, we can see a little bit better the MPAs. So Mabini has the largest MPA in the area, and here we can barely see the MPAs in San Isidro and Mati. They are extremely small. And in each municipality, we looked into 3 different barangays or villages for the data collection. For data collection, we use 3 methods. So first, the MPA Management Effectiveness Assessment Tool, or MEET for short. Second, the semi-structured questionnaires, and lastly, focus group discussions. So the MEET was a tool developed by the Coral Triangle Initiative, and like the name states, it allows us to assess the management effectiveness of each MPA, and it's based on 48 questions that will give us 3 levels of interpretation. So first, each question has a score, and this will give us an overall score of the MPA.
Some questions are considered thresholds from the MPA, so if the MPA achieves certain thresholds, it can go on to the next level. And lastly, we have management focus. This really reveals the weaknesses and strengths of each MPA according to these 9 focuses. So it goes from management plan all the way to site development of the MPA. For semi-structured questionnaires, this allowed us to gather data from small-scale fishing communities regarding their demographics, fishing characteristics, climate resilience, and food security. For the climate resilience, we used 12 indicators based on literature review. These are indicators that enable climate resilience on fishing communities, and this allowed us to insert each of the respondents in one of these categories, so from highly vulnerable to highly resilient for climate change. For the food security, we chose food consumption score method. This was developed by the World Food Programme in 2008, and it's a really simple method where we asked the respondent in the last week how many days did he consume each of these food groups, and this will allow us to insert again the respondent in each of these categories from poor food insecurity to acceptable. Lastly, we used focus group discussions as a qualitative method to further discuss certain topics, such as MPA management and involvement with the communities, and the community's perception of food security. And this was done at the last stage of the study, and it really allowed us to have a deeper understanding of these topics that went beyond the meat and the food consumption score methods. So here we have the type and number of respondents. For the meat, we interviewed knowledgeable people from each MPA that knew about the management of the MPAs, while the semi-structured questionnaires and focus group discussions, we focused solely on small-scale fishers. For the statistical analysis, we used Crystal-Wallace test to check for differences, and we also used spearmint correlation analysis to check if there was any relationship between variables and the strength of these relationships. For results, here we have the first 2 levels of interpretation from the meats regarding the MPAs. Here we can see the overall score. We had pretty good results comparing to the other data that we have from MPA's database. Mabini had the highest overall score with 79 out of 84, and Lupuan had the lowest score with 67 out of 84 points. However, when we look to the effectiveness level, only level 1 and level 2 were achieved, so none of the MPAs were able to achieve thresholds necessary for the level 3 and level 4. Here we see the management focuses. Like I said before, Mabini scored the highest, and so it was expected to also score the highest among the different focuses of the MPA. And we can see that all 4 MPAs scored 100% in legal instrument and community participation, while the main weaknesses were financing, enforcement, and monitoring. During the FGD results, all the fishers across municipalities received several benefits from the MPA. However, only fishers in municipality of Mati revealed they had access to any alternative livelihood programs that were provided by the MPA. When asked about training, fishers stated that they had access to some training, but it was irregular and not access to all of them. And this training was not provided by the MPA, but it was from other agencies such as BFAR, DNR, etc. So now for the small-scale fishing communities, our respondents were mostly male, with an average age between 42 and 46, and we had a big diversity of ethnicities and religions. Most of the respondents stated they have been experiencing more regular and extreme weather events, as well as a decline in fish catch, with climate change being the main reason for this decline across all 4 municipalities. Here we can see the different climate resilience levels across the municipalities. So Lupuan has the highest percentage of respondents being resilient, followed by Mati, Mabini, and Lupuan. While food security, this is according to the food consumption scores, so we actually had pretty good results. Mabini had 99% of its respondents being food secure, according to this method, while Lupuan had 76, which is still a good result. However, the FGD unfortunately revealed a different reality.
All fishers stated that they do eat 3 times a day, however, they do not think it is enough, and in periods where they cannot fish, of course, they all stated that their food consumption goes down. For the statistical analysis, we did find 3 positive correlations. So between the food consumption score and the meat overall score was the strongest correlation, followed by experience fishing and resilience, and age of the fisher and resilience. So are MPAs truly effective? During our literature review, we found that 27.99% of the MPAs in the Philippines did achieve levels 3 and level 4. However, a much higher percentage of the MPAs actually scored much highly in the overall score, and this revealed that a lot of challenges occur in the MPAs across the country, and these challenges were also occurrent in our MPAs. So mainly financing, enforcement, and monitoring financing, because most of the MPAs in the Philippines struggle to be self-sustained. In our study, Mabini was the only protected area that had self-sustaining venues to receive funding. While enforcement in the Philippines is done by Bantay-Dagan, which are local fishermen that are given the authority to conduct the enforcement. And this, of course, will result in bias when the violations occur. And monitoring, because while conducting the study, we did try to have ecological data on the MPA, and 2 of the MPAs had data available. The others, despite the managers stating that they do the assessments, the data was not available for us. So this really reveals one of the main challenges. However, there is a high community support for the MPAs, which is really positive. So all 4 MPAs have weaknesses that need improvement for them to be truly effective. So how are MPAs impacting the climate resilience of small-scale fishing communities? There is an obvious lack of resources from the MPA and from the government agencies. It was clear with core infrastructure when we visited these communities. They are really exposed to climate change. Their housing is extremely expensive. There is a limited reach of alternative livelihoods and irregular training, which was, again, the training that was happening was not provided by the MPA. And so the MPAs have limited impact on climate resilience. So the MPAs enhance food security or create new challenges. We had an overall high food consumption score, which was inconsistent with the FGD results. This could be due to one of the main limitations of the food consumption score, because this method doesn't account for the quantities of food consumption, but it accounts for the diet diversity. And so this is one of the main reasons that we had inconsistent results. And no fishing periods that also affect the fishers. So this area practices closed fishing season from July to September. So during these periods, the fishers cannot practice their only source of income, and this has a similar effect of when it happens extreme weather events. Like it did when I first got there in January, there was huge floods and fishers could not practice their activities. And so despite the correlation that we did find between the meat overall score and the food consumption score, we really need to fully understand food security throughout the year of these communities, and then we can further investigate this relationship. So after these results, we have several recommendations for the MPA management boards. So first, to increase enforcement and monitoring to decrease overfishing. Second, enhance alternative livelihood programs and compensation measures. Third, strengthen local organizations in training. And fourth, improve data sharing regulations between MPAs, fishers, and academia. And hopefully, if the MPAs do these recommendations, it can really bring benefits, both social and ecological benefits, to the MPA and its surrounding communities. Thank you.
Thank you very much.
Thank you.
Sharp within the time.
about fishing, which is the local communities with the small boats that I showed, and
But they
commercial
still sell
fishing.
their fish,
Yeah,
still
they
commercial.
do, but it's but commercial fishing, it's the term they use. Sorry.
Or industrial
It's
fishing.
industrial fishing.
Okay.
Yes. Sorry.
Alright.
So yeah, the regulations are a bit shaky, I would say, and they have a lot of activities that still occur. And even until some time ago, they still use dynamite and cyanide and all these methods. But now they mostly use gill nets and hand lines, so on.
Not an effective. Not an effective.
Not
Thank you.
Thank you.
Thank you, Margarida. So good thing.
Yeah, I would not be that definitive as my colleague and as my esteemed colleagues. I mean, there are lots of examples where MPAs that are no-take zones can be also very efficient. It's all about conciliation between different actors. It doesn't have to be a no-take zone to be inefficient, but there still needs to be lots of enforcement and regulation.
Yeah, that's the main problem that
That was super interesting presentation. I just regret that those slides, you skipped those slides super fast, and I was just no way for me to read, and it was.
Sorry, I was a bit concerned with the time.
Yeah, it was sometimes really fast, and
Yeah,
perhaps
sorry.
you should have tried to show a bit less, but spend more time, based on what we're asking and presenting the NPS, and going like maybe deeper onto like the story that is here. And that's a huge work of passing questionnaires. Did you do it all by yourself?
No, first because of the language barrier.
Yeah.
So we did
But you
have.
did all of them with someone? Or?
Yes, so we did so it was 12 villages that we went to, and we interviewed for questionnaires around 400 fishers. So we had a team, I conducted some of the interviews with a translator, and then depending on the days we had 3 or 4 people helping, either volunteer students or other staff from DNR, which is the Department of Environmental Natural Resources from Philippines.
Okay, that's a huge work.
Yes, it was a lot.
I don't even remember seeing such a huge number of questionnaires being passed for a master thesis. You say that there is strong community support for the MPA?
There is what, sorry?
A strong community support?
Yes, there is.
You never heard anything negative about the MPAs?
We did some fishers. It was a really small percentage of the fishers that said that they do not agree. However, I found that in the Philippines, most of the people have a really strong concept that they understand this is their livelihood, and if they damage the environment, there are not going to be any more fish for them to fish. So they do have this perception, and they do understand that the MPA is going to help them in the long run. So most of them said that they do support, and like I said, the enforcement is done most of the times by the local fishermen. So a lot of the fishermen we interviewed are involved in the enforcement process, and they also help with conducting some monitoring. So it was an overall high support for the MPAs, yes.
Okay, that's super interesting. And you also say that for the 4 MPAs, the fishermen observed an increase in fish catches.
Yes, so the way they said that overall these MPAs, they were established around 2010, 2012. So most of the fishers we interviewed, they have been fishing before the MPA was implemented. And they did state that the decline in fish catch that they had experienced since they first started fishing until now. They clearly see a decline. However, now with the MPAs, they do acknowledge that there is no industrial fishing, there is less destructive methods, and so they do experience a certain increase when compared when they went fishing to other areas that were not protected.
So why isn't the coast of Philippines bordered with a full network of MPAs? I mean, they are the best advocate for this strategy.
Yeah, they actually have a really high number of MPAs, and they are really big on the community management of the MPAs. And they have a high number, however, they don't have the means to actually enforce and to implement these MPAs. So this is what I found when doing the literature review, that most of the MPAs they call the paper MPAs, so they're just
Yeah.
paper and they're not.
But if the fishermen, the fishermen are the best place to evaluate the effectiveness of the
but
MPA.
What we
So
found,
if they observe an increase in fish catches, is that the MPA is working.
yes, but what we did find was, for example, I mentioned aquaculture still happens. When we went to one of the villages in Mabini, actually, the entire area in front of the village was devastated by aquaculture. And when I asked about it, they said it was actually an international company that had came there and placed the aquaculture project.
It's shrimp?
Yes, and so they don't really have that much control because outsiders come. It's same with the industrial fishing. It's mostly China vessels that come and fish in these areas. So even if the fishers do support the MPA, there's still outsiders that come and place these activities. So.
But shrimps are good. I love to have cheap shrimps. Okay, thank you very much. I'm
No one
provoking.
should touch shrimp. Very kind.
That's a provocation.
Except for the European one, even that.
There is still room for one more question. So I will just make you a comment. I really liked your presentation.
Thank you.
Very nice and very attractive, and the topic is indeed very important and relevant for this master. Just point, perhaps you told this viva voice, but I couldn't see the approval by the research ethics committee. Since it involves, this is social research,
Yes.
it involves persons.
Mm-hmm.
So it's compulsory to have an approval by the competent research ethics committee. I'm sure you have it
Yes.
or your supervisor
Yes.
has it, but I'm missing it here.
Mm-hmm. Yeah, I need to discuss it with my supervisor for the
Yeah, it's if you want to publish it,
yes, it's
please
necessary.
be sure that you put the reference number of
Okay.
approval.
Okay.
The reference number or just a statement that you got the approval by the competent
Okay.
ethics committee,
Mm-hmm.
because this is important for social studies and for all studies, as you know, involving research with animals,
Yeah. Thank you.
whether they are humans or not. In the other two cases, it doesn't apply, obviously, for thesis and models, but for in this case, it applies. And then I missed the p values for correlations. You presented the correlations.
I did not present the key values.
I'm sure you got them.
Yeah, no, I got them, but
In the presentation, I missed them.
yeah, I.
Just two very minor points, but
Yeah, okay.
yeah.
Mhm. Thank you.
And okay, that's it. Thank you very much. So now the session is over. So I'm invited to have lunch.
Yeah.